0610_W23_Ms_41. 1 A I Y is FSH. Z is LH. 1 A E. Line DRN. Low until day 14, increasing until day 21, then a decrease. 1 A E 14. 1 A IV placenta. 1 B. Step 1. 0.029 m. Step 2. 75,762. Step 3. 75,800%. 4 megapixels 1 for correct step 1 conversion. MP2 220.029 equals 21.971 millimeters in step 2. MP3 21.971 divided by 0.029 x 100 equals 75,762 in step 2. MP4 correct rounding to 3 significant figures. ECF from previous step. 1 C. Any 3 from flagellum, many mitochondria, acrosome containing enzymes, haploid nucleus. 4 A. Repair, asexual, separate, diploid, zygote, fertilization. 4 B I. Random, change in the base sequence of DNA. 4 B E. Ionizing radiation, named chemicals. 0610_M23_Ms_42. 6 A. Plasmid, restriction, sticky, gene, DNA ligase. Recombinant plasmid, asexual. 6 B any 2 from. Reproduce rapidly, no ethical welfare issues. Ability to make complex molecules. Easy to maintain or store, don't take up too much space. Idea that share a genetic code with other named organisms. 6 C any 3 from. 1 Additional nutrient energy, content may be too small. 2 Seeds may be expensive to purchase. 3 Farmers unable to save seeds. 4 Ref. To side effects unknown, health risks to consumers. 5 Risk of cross-contamination with wild rice. 6 Reduction in biodiversity, genetic diversity. 7 Consumers won't buy GM products, ethical concerns. 6 D. Scurvy, vitamin C, rickets, vitamin D, calcium. 0610_M22_Ms_42. 6 A. Name of enzyme, function site of action. Pepsin breaks down protein to amino acids, polypeptide stomach. Trypsin breaks down protein polypeptides to amino acids, peptides. Small intestine, maltase breaks down maltose to glucose. Intestinal epithelium, small intestine. 1 mark for each correct row. 6 B I. A gene allele. C ribosome. 6 B E. mRNA, a copy of the gene array. Moves out of the nucleus to the cytoplasm. 6 B E T. He sequence of bases in mRNA. DNA. A gene. 1. 6 C. Ref. To specificity, shape of active site and substrate must be complementary. To enable enzyme and substrate to bind, enzyme substrate complexes to be formed. 0610_W22_Ms_4 extended 3. 3 AI 7.6 billion 7 and 600 million sevenths 0.6109. 3 A E 0.3% 2 megapixels 1 correct calculation. MP2 correct rounding to 0.3%. ECF if wrong values used in the calculation. 3 B I. Any 3 from. Named crop disease, war, drought, flooding, unequal distribution of food, widespread poverty, semicolon. 3. MP7 e.g. plagues of crop pests. 3 B E any 4 from. 1 named chemical other than fertilizers used in farming. Pesticides, insecticides, herbicides. 2 kill, harm, non-target species in natural environment. 3 example of specific impact from harm of non-target species. 4 loss of biodiversity, disrupt food chains. 5 bioaccumulation, bioconcentration, biomagnification. 6 ref to resistant organisms or super, bugs, weeds. 7 pollute, destroy, non-target named, area, habitat. 8 antibiotics cause antibiotic resistance. 4 MP1 examples. Herbicides 2, 4D weed killers. Pesticides, insecticides, antibiotics, plant growth. Regulators, animal hormones. MP3 e.g. loss of pollination by bees. MP5A descriptions. MP9 changes, increases the pH of soil. 3BE a single crop in production. 1. 3BIV genetic engineering modification. 4AI habitat destruction, named example. Removing too many of a plant species. Named pollutants of plants. Additional named pollutant of plant. Introduced species disrupted food chains. Disease, e.g. extinction, of other species fewer, pollinators named. Pesticides killing pollinators reduced genetic diversity stated impact. Of GM crops war. 4 any 4 from. More carbon dioxide is being released in the atmosphere. Carbon dioxide is produced by burning use of fossil fuels. Deforestation leads to more carbon dioxide in atmosphere. More methane is being released in the atmosphere. Methane is produced by farming. Methane produced by landfill. Increasing human population. Enhanced greenhouse effect. Greenhouse named gases trap heat infrared radiation. Semicolon. 4 any 3 from. Ensure genetic variation. Represent many environments where the plant grows able to survive. In a changing environment, more chance of cross-fertilization. Not enough seeds in any one population. Increases chances of successful germination. Semicolon. 3 mutations of DNA genetic change base sequence of DNA. 1 any 2 from. Low kinetic energy. For enzymes, low rate of effective collisions. Slow diffusion of oxygen. 4 any 3 from. Water, oxygen, suitable optimum temperature. 4 obtained named resources from larger deeper area. So that they have enough roots to survive the reintroduction process. Ref. To large R surface area. More root hair cells. For more water and named minerals ions salts uptake. Anchorage described, so that not pulled out when animals grazing wind etc. Reduced soil erosion, e.g. named use of water mineral ion. 4 lag. 0610_S23_Ms_41. 5 AIX, nucleus. Y, mRNA. Z, ribosome. 5 A sequence order, a base is in, mRNA DNA gene, 1. 5 E. The sequence of amino acids determines the shape of the protein receptor. Complementary specific shape is required for the receptor molecule to bind, attach, fit to neurotransmitter. 5b. Not all genes are expressed, switched on, activated. Cells only produce the specific proteins they need. 5c. Either an alternative form of a gene. 5d. Humans choose specific features, desired features. Humans choose individuals' offspring to reproduce. Selection not influenced by environment, humans manipulate. Environment decreases genetic variation, decreases size of gene pool. Faster, shorter. For economic, aesthetic reasons. No less. Evolution. E.g. decrease in fitness. 5e any 2 from. Meiosis. Random mating, cross-pollination, crossbreeding. Random fertilization. 0610_S23_Ms_42. 2 a circle 1 pair of the inward pointing horizontal sections opposite each other. 2b 21%. 2 ci gene. 2 ce any 5 from. Ribosomes make proteins, polypeptides. mRNA is copied from gene DNA. DNA stays in the nucleus, mRNA is made in nucleus, mRNA leaves. Nucleus mRNA moves to cytoplasm. mRNA passes through ribosome. Ribosome assembles amino acids, translates mRNA into a protein. Protein synthesis uses energy. Order of amino acids determined by base sequence of mRNA DNA gene. 2 ce any 2 from. Carriers, receptors, proteins. Maltase enzyme. 0610_S23_Ms_42. 3 AI correct readings from graph. -44%. 3 megapixels 1 correct readings from graph 180 and 100. MP2 correct answer to any number of significant figures, i.e. -44.4 recurring. MP3 correct rounding to 2 significant figures. ECF from previous step. 3 any 3 from. Daily doses use of erythromycin peak, 1989, at 2.8 doses per 1,000 people. Bacterial infections resistant to erythromycin peak, in 1993, at 180 bacterial infections per 1,000 people. No record of resistant infections, until 1991, from 1983 to 1989 first 6 years. Delay of 4 years between peak of doses and peak of resistant infections. 3 any 2 from. Fewer doses of erythromycin used. Development of new, antibiotics treatments, vaccines. Any example that would cause a reduced usage of antibiotics. More, awareness education about, overuse of antibiotics, antibiotic resistance. Improved, detection, screening of pathogens to avoid spread. Ref to improved, cleanliness, hygiene or more people. Vaccinated population has reached herd immunity. Isolating infected individuals. MP3 examples. Only giving antibiotics when essential. Do not use antibiotics for viral infections. 3. IV any 5 from. Ref to random mutations occur in some of the bacteria. Genetic variation, inability of bacteria to survive antibiotic treatment. Bacteria with antibiotic resistance survive, reproduce, or pass on resistant gene allele to offspring other bacteria. Increase in frequency of resistant allele gene in the population. Ref to natural selection, become better adapted for the new environment, e.g. gene for resistance transferred to other bacteria in a plasmid. 3. Bi any 2 from. Cell wall. Circular DNA. DNA is free in cytoplasm, not in a nucleus. Cell membrane. Cytoplasm. Ribosomes. Unicellular, single-celled organisms. Asexual reproduction. 3. Be movement, swimming. 3. Ai 3 body regions, head and thorax and abdomen. 3 pairs of jointed legs, 6 legs. 3. Any 2 from. 1 spray use, insecticide, pesticide, chemical to kill aphids. 2 grow crop in, glass house protected environment. 3 use crops that are resistant to insects, aphids. 4 ref to crops that have been produced by selective breeding, genetic engineering. 5 biological control, any example. 6 avoid monoculture, plant a variety of crops, crop rotation. MP5 e.g. predators, parasites, sterile males. MP7 e.g. pheromone, hormone, traps, e.g. surveillance. 3 any 2 from. Upper epidermis, xylem, palisade, mesophyll layer. Spongy mesophyll layer. 3 Bi 252, 1. 3 Be any 3 from. 1 carbon dioxide 14C enters leaf through stoma taught by diffusion. 2 ref to photosynthesis. 3 is used to make glucose, simple sugar. 4 idea of photosynthesis is catalyzed by enzymes. 5 reactions occur in chloroplasts. 6 glucose, simple sugars converted to sucrose. 3 Ci any 2 from. Buds, root, tips, tubers, storage. Organs, flowers, fruits, seeds, young or growing leaves, shoot. Tips, nectaries. 2 any parts of a flower. 3 Ce any 3 from. 1 sucrose is converted to glucose. 2 sucrose, glucose, sugars respired provide energy. 3 any plant process that requires energy, e.g. growth, reproduction, flowering, active transport, absorption of ions, cell division, mitosis, metabolism, fruit formation. 4 stored as sucrose, starch. 5 used to make cellulose for cell walls. 6 converted to amino acids used to make proteins. 7 e.g. used to make nectar. 0610_W19_Ms_43. 5 1 nucleus, chromosomes. 2 bases. 3 protein. 4 genes, alleles. 5 same. 6 sticky. 7 recombinant. 8 bacteria, vectors, viruses, yeast, prokaryotic host cells. 9 insulin. 5 B. Washing powders. Pectinase for fruit juice production. Lactase for lactose-free milk. 0610_W19_Ms_41. 2 carbohydrates. Cellulose. For cell walls. Starch. For energy, respiration. To attract insects to flowers, nectar, fruits. Amino acids. To make named proteins. For enzymes. For growth. 2 B. Correct position labeled on the leaf. Correct position labeled on the stem. Correct position labeled on the root. 2 C I. Higher concentration in the stem, aphid. D is nearer the root, is before the branching of the plant. Sucrose moves by translocation. Sucrose moves up the plant. Root, tuber, is a source. Leaves, stems are a sink. No photosynthesis in the dark. No less glucose, sucrose made in the leaves. Plant uses stored starch from root. 2 C E. Insert gene ref. To genetic engineering ref. To genetic modification. Gene, for insect aphid resistance. Ref. To insecticide described. Description of how insecticide applied biological control grow in glasshouses netting. 2 C E pollination. e.g. biological control described insect products e.g. honey. 7 A. I. Named bacteria. Lightning. 7 A. E. Process A. Denitrification. Process B. Nitrification. 7 A. E. Ammonia. Ammonium ions. 1 A. Nitrite ions. 7 A. IV. Removal of nitrogen-containing part of amino acids to form urea. 7 B. Ribosome. Rough endoplasmic reticulum. 7 C. Protease. Pepsin. Trypsin. 3 E. 1 correct parental genotype A. 2 correct parental genotype A. 3 correct gametes from the parental genotypes. 4 correct offspring genotypes from their gametes parental genotypes. In any order. 5 correct offspring phenotypes and correct probability. 5 megapixels 1 and MP2 parents may be either way. Round but following rows must match. ECF from MP1 and MP2. ECF from MP3. ECF from MP4. Expected answer. Parent 7 man who is heterozygous. Parental genotypes A. Gametes. AA plus AA. Offspring. Genotypes A. Offspring. Phenotypes without with. Cystic fibrosis. Cystic fibrosis. Probability 50%, 1 in 2 over 0.5. 0610_M21_Ms_42. 2 A. Bacterial plasmid, loop of DNA, circular chromosome. 2 A. Ribosomes, cell, membrane, cytoplasm. 2 B. Fungus, protoctist. 2 C. 1 idea of involvement of named bacteria. 2 idea that A is decomposition, decay, deamination. 3 releasing ammonium ions to the soil. 4 idea that A is also nitrification, ref to nitrifying. 5 convert ammonium ions to nitrite nitrate ions. 6 B is nitrogen fixation, ref to nitrogen fixing. 7 by lightning. 8 in root nodules, leguminous plants. 9 idea that C is denitrification, ref to denitrifying. 10 in anaerobic conditions described. 2 C E active transport, 1 A diffusion. 3 A 17 times 0.04 S equals 0.68 S OR 2.8 divided by 4 equals 0.7 OR 4 divided by 2.8 equals 1.43 beats per S. 60 divided by 0.68 equals 88 BPM OR 60 divided by 0.7 equals 86 BPM OR 1.43 times 60 equals 86 BPM. 0610_S21_Ms_43. 2 A mRNA, messenger RNA, 1. 2 A ribosome, 1. 2 A any3 from molecule. A mRNA is a copy of the gene for insulin. Transfers instructions from DNA nucleus to cytoplasm. Ribosome B passes through B. Sequence of bases determines the order of amino acids. 2 B. Insulin is a hormone, stimulates liver cells and muscle cells to absorb, take up glucose, sugar, from the blood. Across cell membranes, stimulates production of glycogen in cells. Reduces concentration of glucose, sugar, in the blood. Homeostasis. 2 C any2 from glucagon, hormone, amylase, trypsin, protease, lipase, semicolon. 4 A any5 from 1 habitat loss, deforestation. 2 large mammals need large amounts of space. 3 named example of climate change, e.g. desertification. Use of land. 4 land, developed for agriculture. 5 land developed for housing, transport, factories. 6 land used for extraction of named materials. 7 competing with humans for space. 8 idea that populations are isolated. 9 hunt trophies, high value poaching. 10 LS are not enforced to protect from hunting. 11 lack of education. 12 poisoning, pollution. 13 need a large quantity of food, lack of food sources, starvation. 14 slow reproduction rate, hard to breed in captivity. 4 B any2 from inbreeding, reduced genetic variation. Reduced variation, less able to respond to environmental changes, disease. Increase in homozygosity described in terms of alleles. Increased chance of genetic diseases, birth defects. Any example, e.g. lameness. Increased chance of infertility. 4 B any3 from 1 collect semen, sperm, implied by context. 2 freeze semen, sperm, use sperm bank, use sperm collected, far. Long ago. 3 selecting, washing, screening, of sperm. 4 use hormones for super ovulation. 5 determine when female is about to ovulate. 6 insert semen, sperm, into female vagina, uterus. 6 AI O I B I AIB I BI O EOI AIOI OI O. Phenotypes of the children AB B A O. Probability blood group O 0 25/25% 1 in 4. 1 mark for 4 correct gametes in Punnett square, 1 mark for 4 correct offspring genotypes. In Punnett square, 6b, both 2 alleles are expressed in heterozygote phenotype. Ref. 2, blood group ABI, AIB, 6c any 2 from. Phenotype is determined only by a gene. No effect of environment on phenotype. Only, 4 limited number of phenotypes blood groups categories. No intermediates between categories. 0610_S21_Ms_42. 5. Helix. 1 the same equal identical maintained constant. Identical. 5b I. 0.003 2 megapixels 1 correct reading from graph IE3 mu m. MP2 correct conversion, ECF from wrong readings in MP1. 5b E. Any 2 from. There are some before division cells that are less than 6 micrometers. The smallest that divide are 4.5 plus or minus 1 micrometer in length. Some cells are less than 3 micrometers after division, assuming equal division. 2 units must be stated at least once. 5 Ci any 2 from. Cut using restriction enzyme. To form sticky ends. Plasmid cut with same enzyme as new gene. To form complementary sticky ends. 5 Ce any 2 from. Rapid rate of reproduction. No ethical considerations. Share same genetic code as other organisms. Ability to make complex molecules. Do not take up much space, cheap to maintain cultures. 3. Transmission of genetic information from generation to generation. 3 BITT. TT. TTTT. 3 BE. Cats. 3 and 4 are homozygous recessive, do not have the allele for polydactyly. 3 CI. Any 2 from. Cats with normal number of toes have AGA for bases 7, 8 and 9. Cats with polydactyly have GGA or AGT. Bases 7 and 9 are different. Base 7 is G nada in the USA cats, base 9 is T nada in the UK cats. 3 CE mutation. 3 CE. Origin of the cat was USA. Base sequence is the same as the other cats from the USA, they have the same mutation base sequence as the Oregon and Missouri cats. 2. 3 D. Semicolon. T, A, AT, AT, T, A, GC, CG, GC, T, A, GC, 1, 3 E. Distinct, phenotypes categories. No intermediates phenotypes not on a continuous scale. 0610_S20_Ms_43. 5. Any 2 from. Nucleus nuclear membrane nuclear envelope. Linear chromosomes. Mitochondrion. Endoplasmic reticulum. Vacuoles vesicles. 5b any 6 from. Resistance arises by mutation. In small number of bacteria. Semicolon. Ref. To a random event not related to presence of antibiotic. Antibiotic kills bacteria that do not have the mutation. Resistant bacteria have no competition. Resistant bacteria reproduce. Pass on, gene allele, for resistance. Natural selection. Method of transmission from one person to another described. Gene transferred to other bacteria of different type in a plasmid. 5 C any 3 from. Prescribe use antibiotics less often. Do not use for viral fungal infections. Make sure people complete the course of antibiotics. Develop new antibiotics. Do not use the same antibiotics for too long. Rotate antibiotics. Use combinations of antibiotics, e.g. isolation of patients with antibiotic-resistant infections, good hygiene to prevent spread of infection, reduce use of antibiotics in farming. 0610_S20_Ms_42. 3. Fur coloring, camouflaged so that not seen by prey when stalking, reflecting heat. Streamlined body shape, improved ability to run fast, reduced air resistance. 3 BI transmission of genetic information from generation to generation. 1. 3 BE 2 of the same letter both lowercase. 1. 3 BE 0. 25/25%. 1. 4. 1. 3 BIV any 2 from. Perform a test cross by breeding with homozygous recessive king cheetah. If any of the offspring of the test cross are king cheetahs, it confirms 17 as heterozygous. DNA testing. 3b anyone from. Compare, morphology, anatomy. Compare, DNA, amino acid, sequences. 3c any 3 from. Hunting, poaching, disease, lack of, food, prey, loss of natural habitat, urbanization, pollution, poisoned carcasses, inbreeding, climate change. 3c any 3 from. Captive breeding programs, local cooperation, education of farmer land users, national parks, conservation areas, protect habitats, legislation, public pressure against hunting, monitor numbers 0610_S20_Ms_41. 4a sorghum 1. 4a feathery stigma, stigma with large surface area, stigma anthers hang outside the flowers. 4b C ovary wall, D ovule, E style. 4b meiosis, reduction, division, haploid, fuses, joins, combines, diploid, fertilization, zygote, mitosis. 4c any 5 from. Gives genetic variation, diversity, refta, alleles, genes, DNA, from different plants, parents. Allows mutations to be expressed. Allows adaptation to new conditions, changed environment. New species can evolve, allows natural selection to occur. Pollen exchange between individuals, cross-pollination. Seeds are dispersed, can colonize new areas. Less competition with parent plant among offspring. Seeds may be dormant. Survival through harsh, adverse conditions. 4d any 3 from. Protein synthesis, transport in the phloem, cell division, mitosis, meiosis, active transport, absorption of ions from the soil, growth, movement, muscular contraction, sensitivity, nerve impulses. 6 a transmission of genetic information from generation to generation. 1. 6b 1 correct use of X and Y in responses for individual 5 and individual 8. 2 correct X allele given for individual 5, XBYB. 3 correct X allele given for individual 8, XBYB. 6b any 3 from. Color blindness is a sex-linked characteristic. She is heterozygous for the gene B flat. She has normal allele B, so has normal color vision, but has passed on the recessive allele B to her sons 5 and 7. She has 2 X chromosomes which have the gene for color vision. Father 4 passes on his Y chromosome. 6b any 2 from. Mutation to give recessive allele B occurred in 3 or in one of her parents, 1 or 2 or her grandparents. E.g. other reasons such as donated gamete. 0610_S19_Ms_43. 3a restriction enzyme 1. 3a sticky ends 1. 3a plasmid is cut with the same restriction enzyme as DNA to form sticky ends region A that are complementary to sticky ends of gene CRY. Reference to base sequences being complementary. DNA ligase used to join plasmid and CRY gene. E.g. formation of recombinant DNA plasmid. 3b gene CRY codes for the sequence of amino acids in toxic protein. DNA gene CRY is copied, transcribed to form mRNA. CRY gene DNA remains in nucleus. mRNA moves from nucleus to cytoplasm, ribosome. mRNA passes through ribosomes. Ribosomes assemble amino acids into protein molecules. Ribosomes make proteins, e.g. order of amino acids determined by base sequence of mRNA DNA gene. 3c to kill, harm, deter caterpillars, insects, pests. Higher yield, reduce losses due to certain caterpillars, insects, pests. Higher quality of cotton, use less insecticide, so less pollution, so less money spent on insecticides spraying, so less risk to workers' health from using insecticides, so less chance of insecticide resistance. 0610_S19_Ms_43. 5 caused by a mutation, change in DNA base sequence of gene for hemoglobin, causes a different sequence of amino acids, so abnormal hemoglobin produced. 5b both parents carry the recessive allele for sickle cell anemia. Both parents are heterozygous. Half the gametes of both parents have the recessive allele. Children who are homozygous recessive have sickle cell anemia. There is a 0.5 times 5/0.25 chance of being homozygous recessive having sickle cell anemia. 5c group of organisms that can reproduce. To produce fertile offspring. 5. C.E. People with sickle cell anemia heterozygotes carriers are resistant to malaria. People with homozygous sickle cell anemia are less likely to survive die of sickle cell disease. People who are heterozygous have one copy of the sickle cell allele, are more likely to survive have selective advantage. Idea that they are more likely to breed. Pass on allele for sickle cell. So increase in frequency of sickle cell allele in population. Selective advantage for sickle cell only exists where mosquitoes carrying malaria are present. Ref. To evolution adaptation to local conditions. 6. AI. Stores contains DNA, chromosomes, genes. Controls the cell. 6. E. P. Endoplasmic reticulum, rough ribosome. R. Mitochondria. 6. B. Catalysts. Starch, maltose glucose, pepsin, liver, neutralizes, emulsification. 0610_S19_Ms_41. 5. AI liquid fluid watery, part of blood, 1. 5. E amino acids, 1. 5. Bi plasmid, 1. 5. Be restriction enzyme, 1. 5. Be cutting opening, A the plasmid, with same restriction enzymes. Forming sticky ends. Idea that sticky ends of human DNA and plasmid DNA are complementary. Reference to base sequences of sticky ends. Correct reference to DNA ligase, e.g. inserting gene sticky ends. Joining splicing, e.g. B is a recombinant plasmid DNA. 5. BIV. Reliable constant supply. Produce large quantities in a fermenter. Bacteria reproduce quickly to make more genetically engineered bacteria. Not dependent on blood donations. Idea that no named health risks. Higher quality of product. 5. BV. mRNA moves through the cytoplasm. mRNA molecules move through ribosomes. Sequence of bases in mRNA determines order of amino acids in TPA for protein synthesis to make proteins.